start by thank you for inviting me to this workshop. It's good to be here, even though it's my first time here in this building, but I can see lots of my former students and colleagues, and this has been a lot of fun. Okay, so I'm going to talk about another invasive species, which is not like Horta. It's a little bigger, you'll see it. It's Sargassum muticulum, and I'm going to mostly to focus on the work we have been doing with Sargassum muticulum for the last few years. Okay, so this is the summary of my talk. I'm going to start by introducing the invader here in the Azores Islands. Not really, just to let you know about it. And I'm going to talk about the phases of an invasion, which is the first phase is the transport and the colonization, if the species can survive there or not. Then the second one is the third here, but the establishment, which means the introduced plants will develop populations that are sustainable and can grow. And then the fourth is going back to the colonization, you have two important things, which is the transport vectors and the abiotic filters, meaning there is ecophysiological conditions for the plant to be there. And then the establishment to develop populations that are stable and can grow. An important thing to look at is what are the demographic traits of the population that result in invasiveness, and the interactions with the biota. So the biotic filters, which are the interactions, competition, and herbivory mainly. And then the fourth phase of the invasion is the landscape spread. And I'm going to talk about the Sargassum muticulum and focusing on all these points. Okay, first of all, just an image of Sargassum muticulum. This was obtained in Rio Formosa lagoon. So inside the lagoon, and this is a plant, and these are the plants that can develop in Sargassum. So it's quite different from Calerpa. It has a tree structure component. The plants can go up to 12 meters in many places. So these are serious structure, 3D structure, with lots of impacts, not only on the other species, but lots of impacts. So what's the problem? The problem is that it displaces other biota, mainly seaweeds and seagrasses, because in the Rio Formosa lagoon, actually, these plants developed in a seagrass meadow. So because they have large canopies, they alter the habitat, they alter the hydrodynamics, lots of sedimentation, they reduce the light to the bottom. There is accumulation of silt that can affect biota. Lots of this can be organic matter, so all the eutrophication problems in the bottom. But on the other hand, they can improve the water quality because they are reducing the sediments and the particles in the water. So the water becomes clearer, so there is positive effects. They provide shelter to many animals, and actually there is often more diversity associated with Sargassum muticulum than the original biodiversity there. And they are a major contribution of food for other animals. So we have to look also to the positive effects. Negative effects like Calerpa, they can clog the fish nets with heavy stuff. The ropes, the propellers can be entangled, like for example here. They can affect aquaculture, whatever. But they can also, and this has been studied, there's a study on this, the large accumulation of Sargassum muticulum on the beach, which is actually very favorable to the amphipods of the beaches. So there's positive effects there. Okay, so let's look at the Sargassum muticulum life cycle. The plants are like this. It's a monoecious species, meaning that both sexes are on the same plant. This can be a good trait for invasiveness. You don't need another plant. The eggs are fertilized at the conceptual surface, and many times the embryos, the small embryos, develop on top of the plant. This is very good for dispersal. And so if these plants can float to other places, when they release little plants which can be more effective and can survive more. So these are the phenology of Sargassum muticum. This is the winter morphology, just short short plants, just the basal part of the plant. In winter, they grow. In spring, they attain the maximum densities. All this depends on where, but it can be in Northern Europe in June. And they have gas vesicles. So these plants develop gas vesicles like this, which are responsible for the floating. Then they lose the gas vesicles, and the fronds kind of get more to the bottom. At this time, there is receptacles. The reproductive branches form. And then the plant releases the big fronds and stays only this basal part attached to the surface over the winter. Okay, so where it comes from? Sargassum muticum, this is Japan, China, and you can see the distribution of Sargassum muticum mainly around Japan, also in China. There is taxonomic confusion, many names to Sargassum muticum. It's difficult to distinguish the Sargassum muticum from other species. There is a need for molecular taxonomy of this genus, so it's not simple. This is being done. There are some publications already about this. So in the original native region, there is a lot of similar species to Sargassum muticum. Some pictures from there. This is actually, we know now that this is the origin of the plants, come from here, which is the Settle Island. So this is, in the native region, is not an aggressive colonizer, not dominant. There is many species that are dominant, but Sargassum muticum is not really the dominant species. This is a study that we did actually and this is important. In Japan, in the original native region, the herbivores that are in Sargassum muticum are different from the fauna of other Sargassum. This is important, we'll see later. And this is the million-dollar question. Why from almost 40 species or around 40 species of Sargassum in Japan, only the Sargassum muticum spread through Northern Europe and Europe? We don't know the answer to this. Okay, so this is the history. Now we're going to the second phase, the transport colonization. So this is the origin, and this was transported to Europe and to North America. In North America, the first record was in British Columbia in 1941, very early on. We know that very early also, because this plant, it's believed to be the vector of introduction is the oysters. So there are very early records of oysters being introduced in North America from Japan. And in the 1970s, both in North America and in Europe, there was a massive die-off of oysters. So the native oysters almost disappeared, and there was massive importation of oysters from Japan. This was it's very well described, and it is probably during that mass importation of oysters that introduced the Sargassum muticum. In Europe, there are records since 1973. Okay? Okay, so when you look at invasion, it's good to know, it's not to do this kind of studies, which is the invasion genetics, or from the point of view of genetics, which is interesting to know how many introduction times. So in Europe, this is not done. Well, I mean, in Sargassum, for Sargassum muticum, this is not done. I'll tell you later why. But there is introduction in the 70s here in northern France, but in the 70s, but in the 80s, there was introduction in the famous Tau Lagoon, where all seaweeds get introduced there. In the 80s, and this is not the same because there was no records of Sargassum muticum along Europe, so it was most certainly a different introduction. Here in the 90s, probably a different introduction too, and the spread. And it's very interesting to know also with this invasion genetics to know what's the level of differentiation between populations, are there connectivities, etc. This is a good tool to study the invasors. So I'm thinking about Colerpa here. And the other question is there natural selection in the introduced zone for certain genotypes? You'll see later that this, for Sargassum muticum, it's not really important because there is only one genotype practically that it's introduced. But it's important to know. We did some kind of microsatellite studies, not really studies, but we spent 2 years trying to develop microsatellites for Sargassum muticum. We couldn't do it. It's very, very complicated. We didn't have enough. So the idea of looking at the genetics of the invasion didn't work out for us. So we gave up this, like a friend also. So let's go to other phase, which is the biotic constraints for colonization of a certain place, geographical zone. So we can look at the ecophysiology of the species. This is very important to understand the temperature, salinity, light reaction to the species, to know if these species can develop there, to do kind of risk maps, for example, as you did it, where the species can go. This is important to do, and probably should do this in Azores Island too. We know that the Sargassum muticum is very good at a wide range of temperatures. For example, here we tested some photosynthetic parameters. This is maximum photosynthesis from 14 to 26 is practically the same. So it's fantastic. In 12 degrees range of temperature, photosynthesis, no problem. And we know that it will do very well at lower temperatures than 14. So it has a wide range of temperature, and this is probably a common thing for seagrasses and for introduced species like the invasors. The optimum temperature growth is 25. It grows at 30, but it survives at 0, and so it can survive the winter and it can grow back in the summer, no problem. Salinity. It's quite good with salinity, even though fertilization stops below 17 to 20, but from here to 32, it's a big range, so it can develop in coasts with near estuaries. The germlings actually can grow in the lab, but down to 5 to 6% of salinity, PSUs, not percent. So it's quite good with salinity too. And light. Actually, the optimal germline production is at 45 microeinstein, which is low. And the compensation levels, this is very important because of the depth range. It's 12 to 32 microeinsteins, which is low. It means that it develops in turbid waters, can go deep also in the sea. So this means that it's very good. The economy between respiration and photosynthesis is quite good. So it can stand low irradiancy. So this is a marvelous species for invasion. Right? So the establishment, which is the next phase that we should look at when we are addressing this kind of invaders. The establishment, as I said, as I defined, is the development of self-sustained populations that can grow and expand. So, and to do this, to understand this, you have to understand the demographic traits of the species and also the biotic relationships, like the competition and herbivory, etc., to understand the establishment phase of this species. So this is the establishment in Europe. I'll come to this later on. It will, just to let you know that it went down the coast and it reached this coast. We are here. I'm coming from southern Portugal. So in 2002, we had Sargassum muticum there, and we started actually studying it at that time. We kind of spotted the first populations, and we followed this introduction here in the southern coast of Portugal. So we're lucky to be there. And I'm going to show you now some data about this, the studies that we did. Before that, let me tell you about the recent publication in Journal of Ecology that look at the genetics of this plant. They used Rubisco, mitochondrial, and ETS markers, which are not very good for population genetics because they could not do the microsatellites as we couldn't do. But they came out with important conclusions, meaning that all introduced plants are really Sargassum muticum. This is not clear because at home it's very difficult to distinguish Sargassum muticum from other species. But no, we have Sargassum muticum all over. This is global studies, both in the States and in Europe. Practically no genetic diversity, so there is no natural selection. It's just one I wouldn't say a clone, but very low genetic diversity, so in the introduced range. So this is kind of a specialized genotype that is growing very well, and it goes very well. The source is most likely the central western Japan, the Soto Island, the Soto Inland Sea. That's the other picture that you saw was taken here. And yes, Sargassum muticum was probably transported with oysters. We don't. Okay, so this is the southwestern coast of Portugal, one of the most wonderful coasts in Europe, maybe in many other places. So this is where we spotted for the first time the Sargassum muticum. The local species, this is a very rough coast. It's open to the Atlantic. And there is these tide pools that develop in the high intertidal tide pools, which are very common there and are, as you can see here, covered by 100% of a native species, which is Schistozeira humilis. Okay? So this is the local species, and this is the same kind of pools when Sargassum muticum develops and occupies 100% of the pool. So this is the way that Sargassum muticum expands through this particular zone in Europe. Very rough, very exposed. In other zones, it goes from sheltered bay to sheltered bay. Here, there is no sheltered bays. It's all exposed, but even though it can go down, it can go through this coast because it occupies high intertidal pools. So it's really a fantastic species if you think about it. Seems I like it. Okay, so some things that we look at it is the timing of the reproduction. When do they reproduce? And we compared with the local species, Schistozeira humilis. This is Sargassum, this is Schistozeira, and you can see these are the moons. So Sargassum muticum has a semi-lunar periodicity and peaks around full and new moon in spring times, right? Full and new moon are spring tides. And these occur in the morning and in the evening. Whereas Schistozeira humilis is, curiously, peaks around the quarter moon, and these are the low tides occur here around midday. So there is a different timing of reproduction of both species. They are not competing in terms of timing. This is the embryo settlement. We studied embryo release and settlement. This is only the settlement. And you can see that if you compare the mid-intertidal pools with the low intertidal zones, these are the middle intertidal pools. So you can see that the settlement is actually much higher in the pools than in the lower intertidal. So this is curious. Here the species is quite well adapted to the pools and uses the pools to spread. In other zones of Europe, this is not so. I mean, this is only in a very exposed coast where Sargassum muticum does this. Looking at the relationship between recruitment and grazing of the germlings. You can see that recruitment is higher in the pools than in low intertidal. So the number of recruits, the mean recruitment here you can see that it's much higher in the mid-intertidal than the low intertidal. And there's differences from year to year. But the interesting thing here is some grazing experiments that we did with cages, open cages and no cages. This is low intertidal, this is mid-intertidal. You can see that the early recruitment survival, so the survival of these small recruits, it's higher in the mid-intertidal than in low intertidal, even though the difference here, which means the effect of the grazers, it's less than this difference, even though the effect of the grazers is higher in the pools. So the grazing effect is higher in the pools, but even so, they survive more in the pools. So they are quite adapted to this pool habitat. Okay, I'm going now into the demographic processes. Let's try to understand what are the demographic traits that are so good and that justify the invasiveness. So we studied the demographics of this Sargassum muticulum in 3 sites, 6 pools per site. And we studied in different situations. In a situation like where the pool was dominated by the original plant, the native plant, Cistozeira humilis, and in pools where Sargassum was completely dominated. And this is probably what happens in the succession of the invasion. Like the Cistozeira pools, when they have a couple of Sargassum muticulum there, what is this demography of Sargassum, and what is the demography of Sargassum when it's completely dominated the pools? So it's we are looking at 2 phases of the invasion, the initial phase and a later phase. Okay? And we use this matrix modeling approach. This is the conceptual model. These are the several classes that we followed for 2 years in several pools. These are R is the micro recruits, the J juveniles, the adults plus gas vesicles, because some of them have and this is also a kind of along the time. Then the adults with fertile adults with gas vesicles, and then the fertile adults with no gas vesicles when they lose the gas vesicles, and the bare old fast, which is the winter form. Okay, so let's look at the results here. So we are looking at population growth rate. So how much is the population? So this means if the population is growing better, the Sargassum muticulum population is growing better here in the Cistozeira pools in the first phase of the invasion, or in the Sargassum-dominated pools in a later phase of the invasion. And it's very interesting. It starts, these are generally lower than these ones. So the population grows better in a second phase of the invasion, which is understandable. The other very interesting thing is that the variability in the first phase, so when it's directly competing with Cistozeira, the population growth rate variability is higher, which is interesting because it tells you about the competition. So when there is competition, there is more variability of the population growth rates. They are generally lower, and when Sargassum finally occupies everything, it grows much better and in a much more stable way. So when it eradicates the native species, it kind of grows much better. This is interesting because of there's this lag phase, and there's this theory about invasive species. Do they have a lag phase or not? If they adapt to the local conditions or not? So there's some evidence for a lag phase in this invasion. Okay, this is the elasticity analysis. I'm not going into the matrix modeling. This is just it lets you know. Let's look at the graphs. This is the time, month, from January to December. And this is how much each phase or each transition, like I said, how much each transition. Let's go back to that. Okay, these arrows, they are transitions. So the probability that the plant has to go from juvenile to adult, for example, or whatever. So this is what we're testing. What are the transitions that are more important for the population growth rate? Okay, this is very important to know when you're looking at to be able to control an invasive species, you have to know this. What is the really important demographic transition, because that's where you want to hit the plant to slow down the invasion or to eradicate it. Okay? So this is a very important study, and we can see that this transition here, it's important all over the year. Very important. And this transition here is just survival of the adults. It's not reproduction, it's survival of the adults. And this is in the Cystozeira pool. So when it's competing with Cystozeira, the plant invests in the survival of the plants that are already there. Okay? And there is also another important thing here, zone of transition. This is in summer, fall, which is the survival of the recruits. So once the recruits are there, the survival of their recruits is very important for the invasion. Okay? When Sargassum dominates the pool, adult survival is still important, of course, for population growth, but also this other one, which is the adults with vesicles, are important here in the fall and early winter. So these actually are, if you want to control Sargassum here, you want to reduce the survival of the adults. This is very important. For example, if you want to collect or harvest the plant, what do you harvest? The fertile plants? Or you just the germlings? Or what? No. For Sargassum, you just go there and take the adult salt. It's not even the fertile ones. You don't have to wait for them to be fertile to improve the effect of your management plan. No, we just go there early summer and take the adults. This is the most effective way to control the plant in this kind of zone, right? You have to do this kind of studies because this varies, I bet, along the Europe or United States. Okay, but actually we tested this. We were lucky because we controlled that population that you saw in the video in Rio Formosa by going there and take out the adults. But we probably were very lucky, but the population just disappeared. It was only one, and it was just on the start when they appeared for the first time in the Rio Formosa lagoon, because we are working a lot in the Rio Formosa lagoon. My students know that. we went there, all of us went there and took the plants out, and it worked actually. There is no Sargassum muticulum now in the lagoon, but it was the first phase of the invasion. So, okay, so this is very important to do. This is an interesting graph, complicated. I'm not going to very much into this. I'll explain you. So this is the survival. This is the importance of survival, growth, and fertility for population growth rate. So, and this graph, this is all the samples that all the data, monthly data that we have from this matrix model. So if the dots were here, it's the growth, the important demographic trait. If the dots were here, is reproduction is the important rate. But now, all the dots, and this is the Cystozeira pools. So in the first phase of the invasion, and this is the second phase of the invasion, the Sargassum pools. So what we have here in the first phase, when Cystozeira is there, it's very important the survival, but also the growth. So you need to survive and to grow rapidly to be able to compete with Cystozeira. Once there is no Cystozeira, is the survival, you don't need really to grow a lot, because if the survival of your individuals is good, that's what you need to continue there. So this is the relative contribution of the demographic traits to the population growth rate, which is really what we are interested about when you're talking about invasive species. Okay, so let's talk about the herbivores now. So what are the importance of the herbivores? There is this enemy release hypothesis for the introduced species, which says that in the native range, this species has herbivores that are specific to this species. And we actually remember the slide before that our study that we are going to publish it now. In Japan, the herbivores associated with Sargassum muticum are different from the herbivores associated with other Sargassum species. So they have specific herbivores on that species, and that's why probably it's controlled. Okay? But when you move one plant, one species to a new range, these guys are not there. They are different, right? So what the animal release hypothesis tells us is that because on the introduced range, the plants there have specific herbivores that are controlling their growth, this one doesn't have anyone, so it can grow more. Okay, so this is basically the hypothesis. Yeah, we can check this. This is the place, the southwest coast, and also in the southern coast, because the plant from 2002 to 2006 went over, and we kind of studied this in the population in the west coast and in the south coast. Okay, so we are interested to look if there are differences between the fauna associated to Sargassum muticum and the fauna associated to other macroalgal. Meaning, in the introduced zone, this invasive has specific herbivores or not, because the other ones have. Okay, so we look at this. This is published now, and also the food preferences choices. I mean, the herbivores in the introduced range, they prefer the invader, or they prefer the native species. So we look at this. Okay, the fauna associated to Sargassum muticum. This is Sargassum muticum. This is in the place where there is only Cystozeira. This is Sargassum muticum. The balls are Sargassum muticum, and the circles are Sargassum muticum, and the squares are Cystozeira. Are the herbivores associated to Cystozeira, and here the herbivores associated to Sargassum. So they look different. In the south, there is a lot more species. These are the fauna associated to Sargassum and many other species. Okay, so the conclusion of this work was that the fauna associated to Sargassum muticum actually was different than the fauna associated to the local species, but this varies a lot. It varies a lot with the species, with the zone, with the site. So there is it's not consistent pattern. They are different, but they are not so different like that. Okay. Other references that look at this problem, actually, they all agree in one thing: is that the Sargassum muticum doesn't have much effect on the faunal communities, meaning that the faunal communities associated to Sargassum muticum in the zones, in the introduced zones, they are not specific, they are not very different. Okay? Yeah, but this is going back to Japan. In Japan, yes, there is differences. There is differences between the fauna associated to Sargassum muticum and the fauna associated to the other Sargassum. I have to run because okay. So we did some food preference experiments with many herbivores, and Sargassum muticum is for giblet, for aplysia, for sea urchins, for hydrovia, amphipods, and all of them together. They are all, Sargassum muticum is among the least preferred food for the local herbivores. So there is no specific herbivores, and the others, they prefer the other species. Okay? This is actually very important, right? I mean, if Sargassum muticum doesn't have very good herbivores specialized on it, so it has an advantage over the other species. That's probably why, one of the reasons why. Yes, we did more food experiments. This is growth. This is interesting because this is relative growth rate of several species with herbivores and no herbivores. And you can see that Sargassum muticum has very good growth, both with herbivores and without herbivores. So it has actually grows much better than all the other species. And this is a key thing. Sargassum muticum grows a lot. And the herbivores, they don't make a difference. Just a little bit, but not much. So it can grow more, it can outcompete the other ones, the other species. Okay, there's another hypothesis. which is the evolution of increased competitive ability. What's the meaning of this? This means that invasive plants in the sites where they invade, they evolve this increased competitive ability because they don't invest, because there is no specific herbivores, they don't invest in anti-herbivory compounds. So this energy that is not invested in these anti-herbivory compounds, it's used for growth. So this is the hypothesis. Okay. And now we went to look at a little bit to this problem. This is recently published. This is complex experiments. I don't have time to tell you a lot about them, but in simple terms, we have this is aquarium. Okay, this is aquariums that we use that were divided by a net. So the water circulates from here to there. Okay, so the water is flowing along the aquariums, and there is a net here. So the water goes from this plant to this plant. Okay, and we can put whatever plants we want here, whatever species, like we put Sargassum muticum or other species, and here we can put Sargassum muticum or other species. Okay, and if we put herbivores here, and the herbivores eat this species, the question that we are looking at, are these guys going to react to that? Meaning, these guys release some kind of chemical cue to the water, and the water flows here, and these guys, the other plants, are warned by that cue, that chemical cue, and oh, there's something, there's herbivory going there. Let's start making anti-herbivory stuff. Okay? We don't have an idea what's the chemical here. This is just to see if there is. And so what we do, we use these plants that are being warned by the chemical cues and feed them with the herbivores. And this is control. And this is fresh food, and this is reconstituted food, meaning that plant meshed with agar, so that we lose the morphology questions, because the herbivores can adapt to the morphology of the plant. So we serve the herbivores agar with that plant. So if these guys react to a warning, to some kind of chemical warning, when you give them herbivores, they will eat less than the control. Okay? Did you get this? It can be confusing. Yeah, okay. So we looked at this. Oops, no. We look at this. That's not what I wrote. So what are the results of this? The results were really that Sargassum muticum doesn't change the palatability, doesn't react to any warnings. Though the seaweeds are on the other compartment being eaten by herbivores, and Sargassum muticum stays the same. Okay, doesn't react, doesn't produce any chemicals against herbivores, doesn't matter. In contrast, which is interesting, when you put Sargassum muticum here, the other species can detect a clue. Okay, so when you put Sargassum muticum here with the herbivores, these guys here, they react producing something anti-herbivory. Okay, so palatability of Sargassum muticum doesn't change, but the native species will change. And of course, between them also, we studied several species. But this is the important thing, is that native species, they actually benefit more than Sargassum muticum from the reduction of consumption due to waterborne cruise. So the native species, they detect the signs and they can react so that they are less eaten by the herbivores. And Sargassum muticum doesn't understand the language of these new sites. Okay? So here there's an advantage for the native species. Okay, let's go to the last, which is the landscape spread. This is the spread in Europe, started here. See, this appeared here, the famous Tau Lagoon. So this is the spread along the time and along Europe. And so it started here, went north. This is a different introduction, but this Sargassum, and this is a different introduction, 1992. But these guys are very similar to these guys genetically. So it seems that there is some kind of favored genotype to go in. So, and this is where we studied 2002, 4, 5, 6, and it went here. And there is no more there because the coast, I mean, we don't have any more information. Okay, so looking fast to the spread rate in Europe, this is a plot that it's interesting. You can do that if you have many data on the species. See, all these dots are some kind of reference to the species, that the species was present a long time, and this is a distance. Okay, so all the references of this species, you put it here on time, and what is the distance of that site from the origin? We know that the origin site was in the northern France, 1976, and the distance from this to all the other sites. So this is all the reports of Sargassum muticulum in Europe. If you take the top ones here, you can calculate the spread rate, because you just use this for the regression analysis, and you calculate the slope, and the slope is the spread rate, meaning that in Europe, Sargassum muticulum travels 67 kilometers each year. So each year it spreads 67 kilometers. It's quite fast. In the United States, in North America, we did the same kind of thing. It travels very, very interestingly, exactly the same. It's 68 kilometers, about the same rate of spread. So it seems this is consistent in completely different systems. So this species is quite good in doing that, spreading. Okay, so conclusions. Sorry, this was fast. So conclusions. What can we conclude about this Sargassum muticulum traits that make him so special, make this plant so special to evade North America and Europe? Transport. The transport is related to the oyster. So, of course, this is the filters for the transport. Is the oyster import? How many oysters are imported? And are there shell treatments or not? In Europe, there are shell treatments for the oysters now that come from Japan that can reduce this transport. What about the Sargassum? Why was Sargassum muticulum introduced and not the others? Maybe, this is an idea, because the embryos have longer longevity than other species, and also they can survive attached to the oysters more than any other species. Okay? Well, you need to study this. Colonization phase. You need to understand the abiotic filters. We know that Sargassum muticulum and all invaders have wide abiotic tolerances. This one has high phenology plasticity. Being monoecious is good because if you have the two sexes there, so you can self-breed, so you can reproduce with yourself. We know that it's very efficient on the use of light. It has low compensation levels, can develop in turbid waters. The establishment of the populations, this has to do with biotic filters and the competition with natives. What we know, this is for the southwest coast. We have different reproduction timing, so it has another timing, so it can reproduce. It doesn't compete in terms of timing of reproduction, and they have higher recruitment in the mid-intertidal pools. That's where they develop that. Okay. And the establishment still in the established. We know that the population growth rate increases with the invasion phase, so it's getting better and better a long time. The variability decreases. Is the survival of the fronds that contribute more to population growth rates. We know that the native grazers prefer the native species, but that doesn't matter because Sargassum grows very well with the grazers, and it grows more than the others. And the palatability we just talked about. So finally, the spread. We need to understand fecundity, how much it reproduces, and how effective is this dispersal. We know that this Sargassum is high fecundity, so it has a lot of reproduction. And we know that it floats because it has the gas vesicle. and it can transport the embryos that are attached to the receptacles. They can be transported along the shore, and that's probably why it disperses so fast. So this makes him a very good dispersal species. And the spread rates are about 70 kilometers a year worldwide. Okay, thank you. Yeah, this is the people that have been working on this subject.
Thank you, Rui.
Okay.